Hello, and thank you for joining us for today's presentation, part of our Agilent Cell Analysis Global Conference. My name is Yama Abbasi, and I'm Head of Business and Application Development within Cell Analysis Division at Agilent. Before I introduce our speakers for today, allow me to share a bit about Agilent and our products for cell analysis. Agilent is a trusted leader in life sciences, clinical research, diagnostics, and applied chemical markets, providing laboratories with instruments, services, consumables, applications, and expertise to enable our customers to gain the insights they seek. AC is one of the newest additions to Agilent Cell Analysis Division, which includes Seahorse, Luxcel, and also Biotech, which was recently acquired by Agilent. Agilent's rapidly growing cell analysis portfolio enables deeper, more reliable insights across a variety of cell analysis applications where investigators and drug developers seek to understand complex cellular environments and interactions, including cell fate, fitness, and function. Our comprehensive cell analysis tools are critically important in various investigative fields such as cancer, inflammation, immunology, immune oncology, and as you'll find out today, for virology and then also cardiotoxicity as well. In this session, we have the privilege of having two speakers from Agilent Cell Analysis Division, Dr. Brandon Lamarche, followed by Dr. Xiaoyu Zhang. Brandon Lamarche received his Ph.D. in chemistry from Ohio State University, where his research focused on the chemistry and enzymology of DNA damage and repair, seeking to understand the ways in which organisms strike a balance between these two opposing processes to maintain their fitness, fuel their evolution, and defend themselves against pathogens. He subsequently worked as a postdoctoral fellow at the Salk Institute, where he studied the biochemistry of DNA double-stranded break repair, with particular emphasis on the types of DSBs generated by cancer chemotherapeutics. He now works as a principal scientist at Agilent Technologies, where he focuses on developing real-time cell analysis applications using xCELLigence platform. Currently, his two primary fields of interest are cancer immunotherapy and infectious disease. Our second speaker, Dr. Xiaoyu Zhang, attained her Ph.D. in molecular and developmental biology from the University of California at Riverside and received postdoctoral training at Sanford Burnham Medical Discovery Institute. Dr. Zhang is currently the senior research scientist at Agilent Technology, specializing in the development of applications for xCELLigence real-time cell analysis cardio-related instruments, leading cardiac safety screening programs and providing technical support for RTCA cardio-related systems. Before I hand over the presentation to Brandon and Xiaoyu, there are a couple of housekeeping notes. There'll be a question and answer period immediately following the talk. And to help us get as many of your questions as possible, we encourage you to type your questions during the presentation. Also, our speakers will be joining the networking lounge after the talk. And if you have further questions, please follow the link to this virtual lounge. And with that, I will hand over the presentation to Brandon and then followed by Xiaoyu.
Thank you, Yama. So today I'm going to be talking about antiviral drug screening and characterization using the xCELLigence RTCA eSight instrument. I'll start off by introducing the eSight directly. This is the newest addition to the xCELLigence line of instruments. eSight is the only instrument in the world that interrogates cell health and behavior in real time using cellular impedance while simultaneously acquiring live cell images. Once I've described these two functionalities, I'll then move to a section where I focus on eSight example data, and I'll do this in three different parts. First, I'll look at tracking viral cytopathic effects using this combination of impedance plus imaging. In the second part, I'll look at applying the assay in the context of antiviral drug research. We'll look at a screening step where you're simply trying to determine which drugs within a library have any antiviral activity. And then I'll move to a characterization step where we take hits from the library and characterize them in more detail using IC or EC50 values. To help explain how eSight measures cellular impedance, I'm showing here one of the specialized plates that the instrument uses. They look like regular microtiter plates, but the difference is that they're electronic. So if you look at the underside of the plate, you'll see that there's a gold biosensor array that's integrated into all 96 well bottoms. To understand what the biosensors are doing, I'm zoomed in here on a single well from one of these plates. eSight provides very weak electric potential to the bottom of the plate, and this causes electric current to flow from one biosensor to the next. If cells happen to be present, they act as insulators, meaning they resist the flow of that electric current. Another word for that resistance is impedance. Now, as eSight applies a constant potential, the amount of current that flows through the system is dependent upon the number of cells that are present in the well bottom, the morphology of those cells, whether they're big or small, and also how tightly those cells are bound to the bottom of the well. It turns out that this impedance approach is an extremely sensitive method for virology assays because a canonical viral cytopathic effect includes changes in all three of these properties. So in a typical impedance assay for virology, we start off with an intermediate or high confluence for our cells. And when virus is added and the cells begin to display a cytopathic effect, they start rounding, attaching to the plate bottom less tightly. This allows for more current to flow through the system. And as that cytopathic effect becomes more severe and cells lyse or detach from the cell bottom, even more current is capable of flowing through the system. The take-home message is this: impedance allows for you to track the full continuum of the cytopathic effect in real time and to do so in a manner that's entirely non-invasive. So in the context of antiviral drug assays, what we're asking is simply, does the inclusion of drug stop or slow this progression? Now, the cartoon I just showed you is very much a simplification. The biosensors are actually designed in an interdigitated array, like this photo is showing. You can see that the biosensors take up about 75% of the well bottom, and there's a central region of the well which is available for microscopy. Over the last 16 years, what we've seen with the traditional xCELLigence instruments is that most users are employing impedance as their primary readout, and they're then using images for confirmation. And so what that typical workflow looks like is that you acquire impedance continuously, and then at key time points, remove the plate, take it to a microscope to acquire images, and then bring the plate back to the xCELLigence instrument for continuous impedance monitoring. While that works, the motivation behind development of the new xCELLigence instrument, the eSight, was that we would be able to do impedance monitoring, but also couple that with live cell imaging and do it all in a single instrument. So the idea here is that you're getting impedance and images all from the same well at the same time. There's zero hands-on time once the assay is started, so you never have to do any sort of endpoint processing. And eSight provides bright field images in addition to red, green, and blue fluorescence images. The workflow for eSight is very flexible. I'm showing one variation of it here that involves seeding cells into the specialized plate, putting the plate into the eSight, which is housed inside a regular tissue culture incubator, and allowing for those cells to attach and proliferate over the first day. At that point, the plate can be removed and virus can be added in the presence or absence of drug, and you're now ready to simultaneously monitor the two outputs. So using the microscopy viewing window, you're going to get your bright field plus red, green, and blue fluorescence images, and using the biosensor array, you're going to get the impedance data. So we've seen what the instrument is and what its outputs are, and let's look now at tracking a cytopathic effect using both of these outputs. So the model system is Adenovirus 5 infecting HEC293 cells. If this is all we used, we would be able to acquire bright field images, which are quite useful. But in order to make the assay more information rich, we can get orthogonal perspectives on the cytopathic effect by using fluorophores. So we're using an adenovirus that expresses GFP constitutively behind the CMV promoter, and we've engineered the HEC cells to express red fluorescent protein that's localized to their nuclei. So one of the first things that we're interested in is determining what the optimal virus dosing is for our drug screening and characterization assays. Here we seeded 10,000 cells per well, and 20 hours later we infected the cells with virus that had been 5x serially diluted. This is the impedance signal represented by a parameter called cell index, and we're looking at that as a function of time. This first data is for the uninfected sample. Impedance was acquired once every 15 minutes, so there are 480 data points here. And one of the benefits of acquiring at such high temporal frequency is that it gives information about cell health and behavior that you wouldn't get if you were sampling, once every few hours. So here in this initial phase, this represents the adhesion of cells to the well bottom. Subsequently, we see a proliferation phase. There's a small blip here. This represents the time point when this plate was removed from the instrument in order to add virus, not to this well, but to other wells in the plate. We see that the impedance signal eventually plateaus, and the reason for that is that the cells have reached 100% confluence at this point. It's worth pointing out that in this real-time format, all of this data is acquired from a single well. So it's very different than an endpoint assay where each well gives you a single data point. So what does this look like when we infect the cells with virus? I'll move from very low concentrations to high concentrations of the virus. So initially, we don't really see much impact on impedance at all. By the time we get to about a 9 million-fold dilution of the virus, we're beginning to see a cytopathic effect reflected by this drop in impedance, and that becomes more severe as the virus concentration is increased. So very clearly, there's a dose dependency to this. I'm representing here this as virus dilution factor, but we can also represent it by the MOI. Ultimately, what we decided was that we would work with a very sort of common MOI for a screening or a characterization assay, and that's that we would work at an MOI of 1, represented here by this aqua trace. In this next slide, I'm going to zoom in to that condition. And what I'd like to address is the issue of what this impedance data actually reflects. So if you're not used to it, you might ask, how do I know that this actually corresponds to a legitimate cytopathic effect? And this is where the imaging capabilities of eSight are very useful. While that impedance data was being acquired, cells in the microscopy viewing window were being used to collect images. And so we can now juxtapose these two different types of data. At 2 hours, the cells are still very rounded and have not yet attached. By 10 hours, cells are beginning to attach and spread out. By 25 hours, we see that the cells are fully attached and have proliferated. By 50 hours, the cells are totally confluent and have packed in very tightly next to one another. By 70 hours, we're beginning to see a cytopathic effect. This 70-hour time point here corresponds to 50 hours post-virus addition. And then as we go out to 120 hours, we see the cytopathic effect is even more severe with cell death, rounding, and detachment. So very clearly, there's a strong correlation between the impedance signal and what's physically taking place with the cells. Now, if we're using bright field alone, this is certainly very helpful. But again, if we include the fluorescence channels, it gives us a different perspective on what's going on. And so it allows for us to transition from this to this. We see the cell nuclei in red and the virus-encoded GFP obviously here in green, increasing in intensity over time. So the benefit is that we don't just get prettier pictures, but we can also pull quantitative information out of those images. If we're using bright field alone, we can track changes in percent confluence over time. But now with the red nuclei, we can count the number of cells that are present over time. And with the green signal, we can do things like count the number of cells that are infected, or if we quantify the intensity of the green signal, we can literally watch the replication of the virus. This allows for us to juxtapose different types of information in the plot like the one shown here. Here again in black, we see the killing response by impedance and its inverse correlation with the signal for the virus-encoded GFP shown here. This just gives a general overview of what it looks like to track viral cytopathic effects using both of these types of output. And we'll move now to applying this in the context of antiviral drugs and a screening phase. So the format here, we switched from using red cells to blue cells, and we also changed the seeding density down to 6,000 cells per well. Here, 24 hours after seeding, we added a mixture containing adenovirus at an MOI of 1, along with antiviral drug at 50 micromolar. Now, in some cases, the concentration, or excuse me, the solubility of the drug limited us, and we were not able to obtain a 50 micromolar concentration. Today, I'm going to just show you 5 compounds. The first 3 of these, Cidofovir, Brincidofovir, and ganciclovir, each inhibit the adenovirus DNA polymerase. The 4th compound, ribavirin, his mechanism of action isn't fully understood. And the 5th compound, stavudine, is an inhibitor of the HIV reverse transcriptase, and we're using that here as a negative control. So here again, we're going to look at the impedance signal. And in terms of timing, we're not going to look at the initial 24 hours of cell adhesion and proliferation. So here, t equals 0 represents the moment at which virus and drug were added. If we look at cells by themselves, they proliferate over this 80-hour window. When we add in virus, it takes about a day, but eventually there's a very severe cytopathic effect. When we include the negative control, stavudine, you can see that it has minimal impact on impedance. The same is also true of ribavirin. But with ganciclovir, cidofovir, and brincidofovir, you see a pretty dramatic shifting in the impedance signal. This is an example of a gain of signal format where the virus is causing loss of signal. And if you have a positive hit, that drug is causing a gain in signal to look more like the cell alone control. This is important because when you have a gain of signal assay, a positive hit is consistent with the drug being effective against the virus and that drug not being toxic to the cells. So if brincidofovir was toxic, we would see the impedance signal dropping, not increasing. The data is pretty clear in its current format, but we can represent it in a few different ways. One of them would be to calculate the area under these curves, as I'm showing here. And this just helps to demonstrate that indeed impedance is very efficient for being able to rank order drug efficacy and determine what is or is not a hit for screening. An important question is whether these impedance data are an accurate reflection of what's physically taking place with the cells. And this is where we can make use, again, of the imaging capabilities of the instrument. So without infection by virus, we see the cells are proliferated. They're highly confluent. When virus is present, there's a severe cytopathic effect, and we see a whole lot of that virus-encoded GFP present. The first 3 or 4 drugs don't really have a whole lot of impact on what we're seeing in terms of morphology or the GFP signal, but cidofovir and brincidofovir obviously suppress the viral CPE pretty dramatically. So the answer to this question is yes, indeed, there's strong correlation between the impedance and the imaging readouts. On the basis of this data, we decided to study brincidofovir in further detail in order to determine its EC50 value. Now, before I show you those assays, I want to address the issue of assay quality. Like, how effective is this assay for being able to detect hits that have activity against adenovirus? When you think about the ideal assay, it really should have a large signal. There should be low variation in that signal. There should be low background, and there should also be low variation in the background signal. People have developed a number of different ways in order to quantify the quality of a screening assay. And one of them that has gained favor in the last 20 years is this Z prime factor. This equation takes into account the standard deviation for both positive and negative controls, as well as the mean value for positive and negative controls. The take home here is that when Z prime is larger than 0.4, an assay is considered to be acceptable or an effective tool for screening. If that value is larger than 0.5, it's considered to be excellent. We wanted to calculate the Z prime factor for the eSight assay I've shown you. And to do so, we took half of the wells in a plate and added cells plus virus plus negative control, DMSO. When we plot all 48 of those data traces at the same time, we can see that here. So again, over the first day, the cells are proliferating, and then a very massive cytopathic effect takes place. In the other half of the plate, we had cells that were infected with virus in the presence of our positive control, which in this case is Brincidofovir. When we plot all 48 of those traces together, we see the data shown here in gray. So again, there's this suppression of the cytopathic effect. It takes a lot longer for this impedance signal to start dropping down towards zero than when no drug is present. We can take these data and put them into the Z prime equation. Here I'm going to plot the Z prime value over time. Reminder, the yellow represents the acceptable range and the orange represents the excellent range for the Z prime value. This is the data. So we can see very clearly that eSight is providing or acting as an excellent tool for the purposes of screening. Now notice that the Z prime score becomes excellent right around 40 hours. Here at 40 hours is when the positive and negative data sets begin to separate out from one another. And in other words, this is the time point where we can begin to have a lot of confidence in the ability of this assay to differentiate between what is and is not a hit. You'll notice that the Z prime score is maximal or optimal at 60 hours. And that makes perfect sense because this is the time point where there's maximal separation between these two data sets. And you'll also notice that the Z prime score begins to decrease over time. And this again makes sense because here the positive control is decreasing in value and is becoming more and more similar to the negative control. And that convergence of those two data sets means that out here at this time regime, it's a less effective tool for telling you what is or is not a hit. This ability to analyze Z prime as a function of time is extremely powerful. You could calculate Z prime using an endpoint assay, for example, at 100 hours, and you would be able to tell that you're getting an excellent score and therefore it's a good tool. But what you wouldn't know from an endpoint assay is that by going to 60 hours, the tool becomes even more excellent. Its power or capacity for determining what is or is not a hit is even better at 60 hours than it is at 100. And so having this real-time data allows for you to make those distinctions. Now we can do the same type of Z prime calculation using, rather than this impedance data, the virus-encoded GFP data, and that's being shown here. Two things to note, the curve shape is largely similar, but the Z prime score from impedance is always larger or better than or more excellent than that determined based on imaging. And the other thing to note is the window over which the Z prime score remains excellent. It's always, it is larger when you're using impedance than it is when you're using the image-based readout. So for impedance, it remains excellent between 40 and 100 hours, whereas for the imaging readout, it remains excellent between 40 and roughly 70 hours. I don't have time to discuss that now, but I would be happy to address that during the question and answer period if anyone's interested. So we'll move now to actually characterizing Brincidofovir, trying to determine its EC50 value. So the assay format remains the same. The difference is that we're now going to vary the Brincidofovir concentration between 8 and 1,000 nanomolar. This is what cells alone look like. This is cells plus virus. And then adding in Brincidofovir from low to intermediate to high concentrations. How do we take this data and determine an EC50 value? Well, again, we can make use of the area under the curve, and I'm showing that here. Area under the curve based on the impedance readout as a function of the log of the drug concentration. So you see this gives us a canonical sigmoidal dose response curve. The R squared is excellent, and it suggests that the EC50 value is around 265 nanomolar. It's worth pointing out that ideally we would like to have some additional data points to represent this upper asymptote. The problem here is that we're limited by the solubility of Brincidofovir. For this particular assay conditions, we just really couldn't get the drug concentration higher than 1,000 nanomolar. So everything I just showed you is based on impedance, but we can do the same type of analysis using imaging readouts. So as we go from no drug to low drug to intermediate drug to high concentrations of the drug, there is a very clear knockdown of that viral CPE. And we can use the eSight software to quantify this. So here we asked the software to encircle all green cells, and then we asked it to quantify the intensity of that green signal on a per cell basis. So I'm plotting that green signal here as a function of time. When cells are treated with virus alone, you see a steady increase in the GFP signal. Around 60 hours, the signal starts dropping, and the reason for that is that the cells are lysing. As we add in drug, we see a reduction in this GFP signal, as you would expect. Now, this is an example of an assay format where there's loss of signal. And it's important to note that there's an inherent ambiguity when you try and interpret loss of signal data. Very clearly, when we go from no drug to drug, there's a reduction in the amount of GFP that's there, but we don't immediately know how to interpret this. One explanation is that Brincidofovir is indeed inhibiting the virus, but alternatively, it may be the case that Brincidofovir is actually inhibiting some of the host cell machinery. For example, if Brincidofovir inhibits the host cell ribosome, then that would suppress the ability to translate the virus GFP's mRNA. So importantly, because the impedance output is a gain of signal, it helps eliminate the ambiguity associated with this loss of signal. That's one of the benefits of having this sort of dual readout. In terms of data analysis, we can again look at this using area under the curve. Here we see the inverse curve shape. Again, the R squared is quite nice. And this suggests the EC50 is around 325 nanomolar. Once again, we would prefer to have additional data points, but the solubility of the drug won't allow us to do that. When we compare the two outputs of imaging and impedance, we see that we have 265 versus 325. They differ by just about 20%, which I consider to be excellent correlation considering that this is a cell-based assay. So with that, I'd like to draw some conclusions. We've seen that using real-time impedance monitoring, we get a very sensitive readout for viral cytopathic effects. And this is being obtained through a gain of signal assay format. We've seen that it functions as an excellent screening tool based on the Z prime factor. And we've seen how we can use impedance to determine EC50 values for drugs that are successful hits from the screening phase. The live cell imaging provides a very nice corroboration of the impedance data. Today, I really only had time to look at the virus-encoded GFP signal, but we could have just as easily incorporated analyses that used the nuclear blue fluorescent protein, the percent confluence, or even included a red fluorescent protein signal that we just didn't make use of today. There's very good correlation between the EC50 values determined by the impedance and imaging outputs. And one of the reasons for that is that both are being acquired simultaneously from the exact same population of cells. This is very different than traditional assays where you might try and run it using two approaches, but they have to be done in different plates and in different instruments. So if the values are so similar from impedance and imaging, what's the value of having both? Well, you have to remember, as we all know, the gold standard in science is that you get a result and then you confirm that result using a different orthogonal approach. Now, unfortunately, because of time and cost, we often don't do that. But with eSight, you get your primary result and your confirmation all within a single assay. So you get at least twice the amount of data, and you don't have to double your workflow. It's the same as setting up a single assay. When we compare eSight with traditional methods like a plaque reduction assay or a colorimetric assay, one of the things to point out is that the EC50 values are very similar between these approaches. And so as one example of that, I'm showing here an analysis of acyclovir's ability to inhibit herpes simplex, different strains. This was done using a plaque reduction assay side by side with an xCELLigence impedance assay. You can see that the EC50 values from these approaches often are just 2 or 3-fold different from one another. So very good correlation. This is one example out of many thousands of papers that have been published over the last 16 years showing good correlation between impedance and traditional methods. Another difference between eSight and traditional methods is how simple the workflow is. You simply are adding cells and virus and drug to the plate, but there's no additional processing or handling of the plate beyond that. And then finally, there's the benefit of having the real-time readout versus traditional assays, which tend to be endpoint. To highlight that, I'm showing here a plot of the virus-encoded GFP signal as a function of time. We see that for cells plus virus in blue versus cells plus virus plus Brincidofovir in aqua. If you happen to be doing this assay in an endpoint format and you chose somewhere around 80 hours for your analysis, you would see that the two types of data are very similar to each other, and you would conclude that this drug is not effective. In contrast, if you analyzed out around 50 or 60 hours, you would see big separation between the two data sets, and you would conclude that the drug is indeed effective. This just highlights sort of the danger of an endpoint assay. Not being able to see the full continuum really puts you at risk of drawing spurious conclusions. Finally, having seen that impedance and eSight in particular can be used for drug screening and characterization, we're now going to move on, and my colleague Xiaoyu is going to show us how this technology can also be used for evaluating the safety of these compounds. I'll be happy to answer any questions at the end of the talk, and for now I'll hand it over to Xiaoyu.
Thank you, Brendan. In my talk, I will discuss the assessment of cardiac safety of antiviral drug using xCELLigence. Drug-induced toxicity leads to drug attrition. Among all these toxicities, cardiovascular toxicity is the major concern. According to this pie chart, the CV toxicity includes not only the drug-induced electrophysiologically changes in the heart, such as QT interval change and arrhythmia, but also the adverse impact on contractility and the structure. Therefore, an ideal in vitro system which could provide comprehensive cardiac safety profile of drugs would be able to evaluate cell responses to them from multiple angles. In order to meet this need, we developed the xCELLigence RTCA CardioECR system. And the system uses this 48-well plate. And the difference between CardioECR system and the RTCA system just mentioned by Brendan is that the CardioECR system includes not only the impedance electrodes to measure the impedance signal, but the field potential electrodes to measure electrical activity of the cells. And again, the impedance electrodes are for impedance measurement. And based on this impedance curve, we can evaluate variability. Besides this information, CardioECR system also can allow us to measure contractility. And based on this contractile or impedance waveform, we can calibrate beating amplitude, beating rate, and the beating rhythm can be evaluated using this parameter called beating rhythm irregularity. And the field potential electrodes measures the field potential signal. And this is a very typical field potential signal you will get from the system. And you are going to get two major parameters here. The first one we call FP amplitude, which is the amplitude of this spike. And the other one is field potential duration, FPD. FPD is the surrogate for the QT interval, which is the EKG readout of our heart. And based on the changes in these two parameters, we can evaluate the ion channel activities of the cell. And here I want to show you the typical data collected from the CardioECR system. And first, we are going to get this time course of impedance readout and the field potential readout. The time scale here could be a couple of hours or 100 hours. So we call this low time resolution data of CardioECR system. And based on this impedance waveform, we can observe cell attachment spreading right after cell seeding and cell growth afterwards. And after we add compound, if we can see the reduction in overall cell index, which suggests the cell death. And the time course of FP readout doesn't really provide much valuable information. However, if you zoom in a data point on this curve, then you will get a high time resolution readout. And here shows the 30 seconds field potential waveforms. And based on the impedance readout, you can get this contractile waveform, and each waveform represents one beat. And since CardioECR system records impedance readout and field potential readout at the same time, the software can superimpose these two signals and show us the excitation-contraction coupling of cardiomyocytes. So in Brandon's talk, he used HEC293 cell line, which is the kidney cell line, for the drug screening, which is a suitable model for his study. However, for my project, cardiac safety assessment, I use this human iPSC-derived cardiomyocytes, which has been widely used as more physiologically relevant cell model for this type of study. In a typical experiment on day 0, we just seed the cells directly into our CardioECR plate, and the plate will then engage in this plate reader, and this plate reader is placed in the incubator all the time, and the cell performance will be monitored right after the cell seeding. And before compound addition, we will do this cell status QC. The reason we are doing that, we just want to make sure the cells can generate very robust and consistent signals before compound addition. So if the cell passed the QC next day, we will add compound, and cell variability, contractility, and electrophysiology will be evaluated from the same cell population. And these are the antiviral drugs tested in this study. No matter whether they are nucleotide analogs or chemical derivatives, they inhibit virus replication via the inhibition of RNA polymerase. And the concentration we tested here, including the concentration below, at, and above Cmax. Except this influenza drug, due to the solubility issue, the highest concentration for this drug was 300 micromolar, which is Cmax. Now let's look at the cardiac safety profile of remdesivir. First, I want to focus on the normalized cell index. As Brandon discussed in his talk, the time course of normalized cell index reflects the changes of cell attachment and variability. At 0.5 micromolar, remdesivir didn't have much impact on cell index curve compared to this DMSO control. However, at 5 micromolar, it started to decrease cell index at 40 hours, which gradually decreased by 60% after 5 days treatment. As the concentration going up, the reduction of cell index started earlier and showed more profound effects. Overall, remdesivir reduced cell attachment and variability in a dose and time-dependent manner. And next, I would like to check contractile activity after compound addition. So in order to do that, I will zoom in a data point on this curve. The traces here show 30 seconds contraction at 72 hours after compound addition. So compared to the DMSO treated wells, the black trace here, the 0.5 micromolar didn't affect contractility, demonstrated by these consistent and robust waveforms. However, at 5 micromolar, we can detect irregular beating rhythm, and at 15 and 30 micromolar, the compound stopped the cell beating altogether. To quantify the observation in contractile traces, we calculated cell beating amplitude, which is the surrogate for the force of contraction as well as the beating rate. The Y values in these figures represent the fold change to the time-matched control, which means we first calculated the fold change compared to its baseline, and then further calibrated to the changes of DMSO control wells at each time point. And the 0.2-fold difference from the baseline was used as the active or threshold criteria, independent of test concentration, because this magnitude of effect exceeds baseline variability. So first, let's focus on the time course of beating amplitude. Overall, remdesivir induced a reduction in beating amplitude in a dose and time-dependent manner. At 72 hours, 5 micromolar remdesivir started to show negative impact on beating amplitude, which was further reduced by 80% at the end of the treatment. And 15 micromolar and 30 micromolar immediately shut down the cell beating activity. But interestingly, 50 micromolar also showed a transient recovery of beating activity from 24 hours to 48 hours post-drug. So like other RTCA systems, CardioECR systems mirrors the cellular events in real time. Therefore, we won't miss any information, even very transient changes during the treatment. This also prevents misinterpretation if you collect the data at the wrong time points in your endpoint assay. So now let's look at the time course of the beating rate. We can see a very similar pattern in the remdesivir-induced reduction of beating rate shown in this figure. As shown in this 30 seconds contractile activity, it clearly shows that 5 micromolar cause irregular beating rhythm. So in order to detect this type of event automatically and mathematically, the BRI, beating rhythm irregularity, is introduced to the software. The BRI is defined as the coefficient of variation of beating period of each individual beats within this measurement. So based on our empirical data, if the BRR value is above 0.1, that will suggest the irregular beating rhythm or arrhythmia. So from this time course of BRI, we can see 5 micromolar remdesivir disrupted the cell beating rhythm after 72 hours post-drug, which remained on and off until the end of the test. So next, I want to discuss the impact of remdesivir on the electrical activity of the cells. So these are two representative field potential signals obtained before the drug addition, the black trace, and 30 minutes after drug addition, the orange trace. These individual waveforms within the measurement were further averaged into a single waveform. And it clearly shows that the FP amplitude was significantly reduced by remdesivir. And the time course of MP amplitude also demonstrated that remdesivir reduced FP amplitude in a dose-dependent manner. The decrease in FP amplitude may suggest inhibition of sodium channel activity. However, we cannot ignore that the cell variability and especially cell attachment also play a very important role in the modulation of FP amplitude. So according to our observation, the reduction of cell attachment can also reduce FP amplitude. Therefore, in order to get conclusive results on sodium channel activity, it's very critical to monitor the cell attachment while the FP signal is being measured. And our CardioECR system does have this capability. And due to the large variation of changes in FP amplitude observed in the long-term experiment, so in this talk, I will only focus on the drug effect on FP amplitude within the first 1 hour after treatment. And indeed, the remdesivir also dose-dependently reduced FP amplitude during the first 1 hour treatment without having significant impact on the cell attachment, as shown in this overall cell index curve over here. And as I mentioned, IP is another important parameter. So here, we also evaluated the few potential duration at 30 minutes post the drug. The concentration at Cmax 5 micromolar and below had no impact on the FP signal, as shown in these blue bars. However, at 15 micromolar and 30 micromolar, they stopped cell functional activity altogether. No contractile and electrical signals were detected. So we call this cell quiescence. So here, we use these two black bars with value of 1 to represent that all replicates tested at these two concentrations cause cell quiescence. And this is the drug for influenza treatment. So according to the data collected from the CardioECR system, it didn't show significant change in cell variability and contractility, including beating amplitude, beating rate, and beating rhythm. And there was also no impact on electrophysiology, which was evaluated by FP amplitude and field potential duration. So overall, this drug appears to be safe in vitro. Like the influenza drug, this anti-hepatitis C virus drug seems to be safe in vitro as well. Even though we detected increase in the beating amplitude 4 days post-drug and the reduction of IP amplitude from 10 minutes to 30 minutes post-drug, these changes are very transient and also on the border of the threshold for cardiac active compound. So when you look at the people around us, you may notice that more and more individuals these days are taking multiple medications, which increase the possibility of drug-drug interaction. Drug-drug interactions poses a difficult drug safety problem. Especially for cardiovascular drugs, there is a long history of drug-drug interaction. Some statistical models have been reported for predicting potential drug-drug interaction with electrophysiological effects. However, no in vitro model has been demonstrated. So during our study, we also asked ourselves, what will happen if a patient take antiviral drug and cardiovascular drug at the same time? And can the CardioECR system, together with human iPSC cardiomyocytes, be used for in vitro drug-drug interaction study? So in order to address these questions, we recruited 2 cardiovascular drugs shown here. They were co-treated with the antiviral drug respectively. So before we performed drug combination study, we checked the cardiac safety profile of these 2 cardiovascular drugs. So from these figures here, we can see that at the tested concentration, 0.3 micromolar for amiodarone and 6 micromolar for losartan, they didn't show cardiac side effects. So now I'm going to show you the results on drug combination test. As I have showed you before, this HCV drug alone had very safe profile. It didn't affect cell variability, contractility, and electrophysiology. When it combined with this losartan, it also appeared to be safe. However, when we combined it with amiodarone, it started to show the impact on cell variability and attachment demonstrated by the decrease in the overall cell index. Also, it affected contractility, which is reflected by the dose-dependent decrease in beating amplitude while increase in the beating rate. In addition, it showed dose-dependent reduction in the field potential duration. And this in vitro data collected from CardioECR system is comparable to the in vivo data. So in summary, xCELLigence RTCA CardioECR system has both field potential electrodes and impedance electrodes, which provides comprehensive assessment of antiviral drugs. And using CardioECR system together with human iPSC-derived cardiomyocytes, we demonstrated that remdesivir impacts cell variability, contractility, and electrophysiology, while that influenza drug and HCV drug show minimal impacts. And this HCV drug, which appears to be safe by itself, does show profound cardiotoxicity when combined with amiodarone. So with that, I would like to conclude my talk, and thank you very much.
Looks like we might be having an audio issue. So I'll go ahead and try and start addressing a couple of the questions that have been posed. So one question was regarding the flexibility of the eSight assay in terms of antiviral drug screening and characterization. And so I showed a format where we add cells to the plate and allow them to attach and proliferate over the first day, and then add virus and drug. But variations of that is that you can use adherent cells that have just been trypsinized and mix them with virus, allow that infection to occur in suspension, and then add that mixture to the plate. When you run the assay in that format, you will get a different impedance profile. The traces look different, but that format can also be used for identifying what is or is not an antiviral, and even for characterizing in terms of an EC50 value. It's also worth noting that it's possible to use impedance for cell types that are not naturally adherent. In order to have an impedance signal, the cells need to interact with the gold biosensors in an intimate way. And so to facilitate that, what we do is pre-coat the well bottom with antibodies that are specific to a protein on the surface of those cells, which are not naturally adherent. We've used this approach extensively in the context of cancer immunotherapy, and it worked quite well. And we actually sell, I think it's 5 different kits that allow for this approach to be used. So in the context of virology assays, that type of approach could also be used. Yama, are we still having audio issues? It seems like we might be. So why don't we do this? I'll go ahead and I'll field a couple of other questions, and one for Xiaoyu. So are the contractility and electrophysiology measured from individual cells?
Thank you, Brandon. So this is actually when you use the CardioECR system, we do have two sets of different electrodes, as I mentioned during my talk. So we have impedance electrode and field potential electrodes. The impedance electrodes measure the average signal from the entire cell monolayer, so not individual cells. For the field potential electrodes, we do have two field potential electrodes, but it's still not measuring the individual cells. It's measuring the cells sit on top of the field potential electrodes or around. So, but the signal is local. So there are difference between these two signals. Impedance is the average from the entire cell monolayer, and field potential is local information from the cell culture. Yeah, thank you.
Xiaoyu, another question is, can the CardioECR system be used for measuring or studying cells other than cardiomyocytes?
Yes, so that's why we also call it RTCA, right? So as other RTCA systems, CardioECR system also can be used to measure other cell lines. However, I mean, that's going to be overkill, because for the CardioECR system, we have a very high data acquisition rate, which is 2 milliseconds for the entire plate. I think, Brendan, the system you're talking about, it's about maybe 15 seconds per data point. So yeah, that's very kind of relatively low data acquisition rate. So that's why the CardioECR system mainly used for contractility. And but again, it can be used for other cell line other than the cardiomyocytes. Yeah.
Okay, great. Another question regarding eSight is how many plates can the instrument accommodate? So eSight can accommodate 5 different 96-well plates. All 5 of those plates can be interrogated using imaging, while 3 of those plates can be interrogated using both imaging and impedance. Another question regarding eSight is the kinetics and what the limitation is. So as Xiaoyu mentioned, for impedance measurement, you can acquire data from an entire 96-well plate in about 15 seconds. It's actually a little bit faster than that. So it's possible to do very rapid kinetics from an impedance perspective. In terms of imaging, the fastest I've ever gone is about 1 image per minute. I think it can go a bit lower than that, but one of the challenges there is the issue of phototoxicity. So whether it's eSight or any other imaging system, the more frequently you're blasting cells with light, the more problems you're going to have with toxicity. And that's also very cell type dependent. So in my hands, I know, like, I've imaged A549 cells over the course of multiple days, where I've collected images every few minutes, and they've done just fine. And then with HEC293 cells, I found them to display much higher sensitivity to phototoxicity. This also will be dependent on which wavelength of light you're using and what the overall exposure duration is to that light each time an image is being acquired. So there's a lot of different factors there. But in short, impedance is non-perturbing, non-toxic, and it can be measured much more rapidly than is the case with images. So for Xiaoyu, another question was, can the CardioECR plates be reused?
Yeah, we heard about that a lot. So no, actually, especially for CardioECR plate, because the electrodes, they are very, very sensitive. We already know during the cleaning process, that's going to damage the electrodes. There are a couple of reports actually from our users. They were saying if you reuse the plate, then you will see a large standard deviation during your assay. So we don't recommend you reuse the plate.
Okay. So another question regarding eSight was whether the assay format would allow for you to include adsorption and washing steps. So again, the assay format is extremely flexible. So yeah, you could imagine having an adherent cell line already attached into the electronic plate bottom, and then allowing an adsorption step with virus that, maybe it's, 20 minutes or an hour, whatever it may be, and then washing the cells and then adding in fresh media on top. Absolutely, you can do that with eSight or with any of the xCELLigence instruments. Now, whether or not that'll have an impact on the impedance signal will vary on a case-by-case basis. So I showed you an example where the plate was removed from the instrument, and that small temperature fluctuation during that time resulted in a transient sort of blip in impedance. You may see something like that, but if the cells are in the same type of media during the adsorption step and then after the washing step, then in theory, the impedance signal should remain constant. Again, temperature may cause a slight fluctuation, but the value should return to where it was prior to the wash step. You have to be obviously very, very careful during washing that you don't dislodge any cells. If you do, that will have a an impact on the impedance signal that will be lasting. But in general, yeah, those types of things are easily accommodated. A couple more questions here for Xiaoyu. Do you coat the plate prior to seeding your cardiomyocytes? And then if you do that, does a coating impact the impedance or the electrical field potential readings?
Thank you, Brandon. Yes, we do have a special coating solution. That's actually just kind of a normal kind of substrate people use for cardiomyocyte, which is fibronectin. And some of our users also like to use matrix gel, maybe applies to your experiment. But matrix gel, you have to be careful because the thickness of the thickness of the matrix gel is actually going to affect the signal collection. Yeah.
Great. So another question regarding eSight is this Z prime factor that we mentioned in terms of quantifying assay quality for hit detection. And the question is, why does that Z prime factor differ when you compare the impedance trace versus the virus-encoded GFP trace? And the reason for this is that it has to do with, at the end of the day, the ability to differentiate between a positive and negative control. So if let me back up and say, let's say that I monitor this process of the demolition of a building, and I record that from 1 mile away using an audio recording and then also a video recording. You're watching the exact same physical process take place, but those two types of recordings are going to vary slightly in their kinetics, based on how fast light travels versus how fast sound travels and that sort of thing. So with eSight, when we track the exact same process, this virus-mediated killing, and we do it with impedance and we do it with imaging, even though they're looking at the same thing, their inherent sensitivities and the kinetics that they're detecting are going to vary a little bit. And what we see is that first, they differ in magnitude of the Z prime factor, and then also the sort of window over which that Z prime factor remains excellent. So let's talk about magnitude. The fact that the magnitude of Z prime is higher based on an impedance readout than it is based on an imaging readout tells us that you can more clearly distinguish the positive and negative controls using impedance than you can using imaging. And the reason for that is that the impedance readout is a more information-rich signal. It includes changes in cell number, changes in cell size or morphology, and then also changes in the strength with which those cells are interacting with the plate bottom. And so because you're looking at so many things together, the ability to detect cells that are undergoing a CPE versus ones which are not ends up being more sensitive than if you're just tracking how much of that virus-encoded GFP is around. And then in terms of the duration of excellence over which that tool is useful, what the data tells us is that impedance remains robust over a longer window, is essentially what the data tells us. I'm kind of struggling a little bit, but in short, impedance is just a more information-rich perspective on what's going on in terms of this hit detection. So with that, I think that we've covered all the questions in the queue. And
I am back, Brandon.
Oh, great.
But obviously, you guys were doing absolutely fine without me, so that's great. Did I miss anything?
Well, we covered the questions in the list here.
I saw those. Yeah, thank you very much for moderating. I had some technical issues. My sincere gratitude to both you, Brandon, and also for Xiaoyu for sharing your work, and also thanks to our audience for the questions and participation. Thank you very much.
Thank you.